Hi everybody, and welcome back to your second laboratory meeting and your second series of AV lectures. In this series we are going to be breaking it into three different units. AV Lecture 2A, AV Lecture 2B, and then 2C. Each of these AV lectures is going to correspond to three labs that we're going to be covering. Labs 7, 8, and 9. Lab 7 is going to be the integumentary system, also known as really the skin. Then we're going to do an introduction to the skeletal system, and then a very specific part of the skeletal system called the appendicular portion of the skeletal system. So let's just go ahead and begin to talk about the integumentary system. And I will be the first to tell you guys, I love the integumentary system. It's one of the most fascinating body systems, and I really wish that we had 16 weeks to talk about it, because there are so many cool things and so many interesting facts and just fun things to discuss with it. But I'll try and bring in a couple cool ones here or there, and then when we meet in lab, we'll discuss more. The skin really is considered the largest organ of the human body. There is a number of associated structures in the integumentary system that we'll discuss, such as hair, nails, and a variety of glands. Things like sweat glands, oil glands, so forth, that we'll discuss. The skin has a whole wide range of functions. We have just, this is obviously not a comprehensive list, and we could make this go on and on and on, but some of the key functions that we'll discuss with the skin are the process of thermal regulation, really protection, really responding to sensations, and then something called vitamin D synthesis. So let's break these down just momentarily. What do we mean by thermal regulation? Well, you all know that the body is set at a normal internal body temperature, and oftentimes it's taught at 98.6 degrees Fahrenheit or around 37 degrees C. Really, the skin functions to help maintain that internal body temp. And how do we do that? Well, we can really play with the vascular supply that's found within the skin, or we can actually undergo excretion via sweat. When we actually are talking about protection of the skin, what are we protecting it from? Well, a variety of things, such as UV damage or UV light that could damage our DNA found within our cells. The skin is considered also part of our first line of defense, meaning out in the world, which our naked eye can't see, we have lots of microbes, lots of pathogenic agents that can cause disease. The skin actually allows us to block off and create a barrier, if you will, from those potential agents. The skin is also going to prevent water loss. Having that integumentary barrier there allows the water to stay within the body's tissues rather than just them losing it. As I said a little while ago, the skin responds to sensory input, sensations, such as touch, pressure, pain, temperature, and even emotional responses. You can tell when somebody potentially gets angry based on really their skin becoming red. Or when they're ill and it becomes very pale. So these are all, or even nervous, could be pale. So different types of sensations that, the skin can really respond to. And then we have vitamin D synthesis. Vitamin D synthesis is really important because it's synthesized in the presence of sunlight. This is why they say you need about 30 minutes of sunlight, a day. What they don't really tell you is that almost all foods today are fortified with vitamin D. So you could pick up orange juice, milk, so forth, and it'll say vitamin D fortified. Well, really, what is the importance of vitamin D? Vitamin D actually plays a role in calcium absorption from the digestive system. Now, this is not really a biochemistry class, and in lecture you're going to talk about the biochemical pathway of vitamin D synthesis in much greater detail. So I am being somewhat vague on this, but Dr. Keller or Dr. Perotti will talk about this quite a bit. Interestingly, all of us, may have had a soda or two in our life, and some of you may be avid Coke or Pepsi or Dr. Pepper drinkers. There is an acid called phosphoric acid found within dark sodas, and what this phosphoric acid does is it blocks calcium absorption. Meaning you could have all the vitamin D present in the world, and you could have eaten 10 gallons of vanilla ice cream or 10 gallons of yogurt, and you've got all the calcium present in the digestive system, but let's say you had a soda right before. That phosphoric acid is greatly going to inhibit the uptake of calcium coming in through the GI tract. There is just sort of a unique couple things to build on that. Is most root beers, not all, but most root beers do not contain phosphoric acid. For instance, I think IBC, Barks, maybe Mug, I'm not entirely sure, but majority of root beers do not contain phosphoric acid. There are a couple that do, but you can see that on any of the ingredients of the back. There have been some researchers that have suggested that children who have had a diet high in dark sodas at an early age have early onset or may have early onset of osteoporosis as a result of blocking calcium uptake, and therefore, bone development doesn't occur as readily. Excuse me, I got a tickle in my throat. All right, we can break down the integumentary system really into two main regions. The first region is the most superficial region that we'll discuss, and that's called the epidermis. The epidermis you see here in red. And then just deep to the epidermis is something called the dermis. The dermis here is this region. The epidermis and dermis are the two regions of the integument that really are anchored by something called the hypodermis. Hypo meaning below the dermal layer. The hypodermis is not considered a layer of the integumentary system, but it does have a couple very important relationships with the integument, such as anchoring the dermis and epidermis down, and it is highly vascularized, meaning there's a lot of blood supply within this hypodermis, and even some of that blood supply protrudes into the dermal tissue. This is going to be important for supplying nutrients to really the dermal cells and the living cells of the epidermis. One other unique thing to talk about with the hypodermis is the site of sub-Q or subcutaneous injections. Really, why is that important? Well, if we can directly access the vascular supply, meaning the blood vessels found within this hypodermis, we can immediately administer maybe a drug to really the body and have it begin to act almost instantaneously. All right, let's go ahead and now talk about the epidermis in greater detail. The epidermis is really comprised of 5 main strata layers, because strata meaning layer. These layers go from really a deep portion of living cells to a superficial portion of dead cells. And if you recall from the histology unit that we talked about, the integumentary system is made up of something called keratinized stratified squamous epithelium, and this is the classic example of it. So we're going to discuss each of these strata layers in greater detail in a moment, but just a couple quick things to point out. One, the epidermis is considered avascular, meaning there is no blood supply within the epidermis. And we have a variety of very, very sparse nerve endings. Most of our nerve endings are going to lie deep, or most of our really nerve accessory structures are going to lie deep to the epidermis. What are those 5 epidermal layers? We're going to start at the deepest, at the basal layer. That's the stratum basale, and you see it right here. Then we have the stratum spinosum, granulosum, lucidium, and corneum. So we're moving from that basal layer superficially. this is a little mnemonic that I like to give everybody. There's many different mnemonics, some of them which are a little bit more inappropriate than this, that you all, if that works in your head, you guys can go Google those. Some of them kind of get pretty raunchy, but if it helps you memorize the stuff, whatever it takes to get the information in your head, go for it. But the mnemonic that I have here is, can little girls or can little guys shoot baskets? And this is going from the superficial portion of the epidermis to the deep. And that's the corneum, lucidium, granulosum, spinosum, and basale layers. So if mnemonics work for you, go ahead and use them. You'll see quite a few throughout this class just because that's one of the traditional study aids that most anatomists give students when you're beginning to learn this stuff. Here is another histological section from University of Wisconsin's Med School. What you're looking at here is, this is actually a beautiful histological section. You have the stratum basale, and the stratum spinosum, stratum granulosum, stratum lucidium, and then that dead layer of the stratum corneum. And we'll discuss those now in detail. The stratum basale, this is the deepest layer, is a single layer of cuboidal-like cells, or in some parts can be small columnar-like cells attached to a basement membrane. Okay? The cells in this stratum basale are actually living, meaning these aren't dead cells, they're actively undergoing cell division, that process of mitosis. What are the two predominant cell types we see in the stratum basale? Well, one, keratinocytes, and then two, melanocytes. The keratinocytes are cells that produce that keratin. That's that protein that we see actively up here that hardens those dead cells and makes it really nice and tight and waterproof. That keratin protects against wear and tear, and then those outside, pathogenic agents that we talked about a little while ago. The melanocytes produce something called melanin. It is a protein pigment that allows for skin coloration or skin pigmentation. This melanin offers UV protection. Where do we see it? Only in the epidermis. Now there are some modified melanocytes found elsewhere in the body, but predominantly we see melanocytes and melanin production being produced in the epidermis. When melanin is produced by the melanocytes here in the stratum basale, the melanin actually then gets put into the keratinocytes that are migrating superficially. What does this mean? Melanocytes stay in the stratum basale. The keratinocytes are the ones that migrate from the stratum basale to the more superficial layers. Well, if melanin is being produced, we want it to actually get put into cells that are going to move in a superficial fashion. Therefore, the melanin is produced in the melanocytes. It gets put into the keratinocytes. Those keratinocytes move up to the superficial portions of the epidermis. Now, just something to emphasize this point. For those of you that have ever tanned, I don't care if it's a tanning bed, I don't care if it's Rocky Point, if it's California, if it's spring break, if it's here at NAU. If you have ever done any tanning, I oftentimes ask students, when does your tan look the best? And the answer is oftentimes really 2, maybe 3 days after the tanning session. Why is that? Well, when these melanocytes are producing melanin because, hey, you're tanning, you've got a lot of UV light coming down in, hitting these cells, and they're going, hey, we need protection from that UV light. Melanocytes here in the stratum basale make lots of melanin, put it into the keratinocytes. It takes time for those keratinocytes to migrate superficially. Well, by the time it's 2-3 days out of that tanning session, the keratinocytes have moved in a more superficial fashion. They're now closer to the surface. Therefore, hey, your tan is quite even and nice. So understanding the stratum basale is really important for that. All right, the second layer that we're going to discuss here is the stratum spinosum. It is the second layer of the epidermis. The second layer that's, let me start that over. It is the second layer deep in the epidermis here. And really it's comprised of several layers of keratinocytes. So you'll look here, compared to the basale layer, it's much thicker. Okay? Deep portions of the stratum spinosum can still be living and undergo mitosis. So this allows more keratinocytes to push forward and allows more melanin to be phagocytized and put in them. We do see mitosis ceasing, though, at the most superior portion of the stratum spinosum. But when mitosis ceases there, we also see more keratin filaments produced. This is where now, okay, cool, the keratin can really begin to work to begin to harden and create that water-type barrier. Third layer that we'll look at is the stratum granulosum. Stratum granulosum is 3 to 5 layers thick of flat keratinocytes. That's what they're showing here. Usually, you will see thicker portions of the granulosum in thicker parts of the body. It's just, you know, some texts go into that, others don't. Just, you don't take it as a rule of thumb, but in some portions of the body, it'll be more than these 3 to 5 layers thick. What's interesting about this layer is that we do see the staining, actually, of these cells much darker compared to the basale and spinosum layer. So one of the ways that I tell students to memorize these layers when they look at the histological section, look at the 2 light layers, and then the immediate portion that you see darkened here, that's the granulosum layer. What do we see in this portion of the epidermis? Cells are going to contain a lipid-rich substance. This lipid-rich substance allows for waterproofing, and it also prevents the skin from dehydrating. This lipid-rich substance, though, does cut off more superficial strata from the nutrient supply. So regions 4 and 5 here, they can't get things like oxygen. They can't get things like glucose and other nutrients that are needed to sustain really that cell's, I guess, metabolic activity. AKA, the cells from here on out are all considered dead. So lucidium, here we have a dead layer of cells. It's a thin translucent zone, and we only see this stratum lucidium in the palms of our hands and the soles of our feet, meaning thick-skinned areas. Typically, it's a little bit lighter or a clearish region in comparison to the granulosum and this top layer, the stratum corneum, which we haven't talked about in detail yet. The cells, because they're dead in this layer, have no nuclei or visible organelles or organelles in general. And then the last layer, the stratum corneum. Stratum corneum is a completely keratinized, hardened superficial layer. It can contain up to 30 layers of dead, really, keratinocytes. It is constantly shedding or sloughing off these cells, and really these dead keratinized keratinocytes have no nuclei or organelles present. A couple cool interesting facts. The entire epidermis will renew itself approximately every 6 weeks, and then about 1 pound of corneum is sloughed off per year. All right, now that you have an understanding of the epidermis, we can go ahead and talk about the dermis. The dermis is really the deeper region of the integumentary system, and you'll see some wavy boundaries here that we'll talk about that are important with respect to the epidermis, and that's what's going to distinguish it from the epidermis itself. So the dermis, one of the main functions is it offers structural integrity and actually support. And what is it comprised of? Really dense, irregular connective tissue. And if you recall from or histology AV lecture, remember dense irregular connective tissue had all those protein fibers that are just randomized. They don't go, they're not in parallel fashions like what regular connective tissue is. You see it in irregular fashion. This allows for structural integrity, much like what the function of the dermis does. We also see some blood vessels, some nerves, some sensory receptors such as the Pacinian corpuscle, Meissner's corpuscle that we'll talk about. And then we have hair follicles and a variety of exocrine glands that I'll discuss. All right. The dermis is broken down into 2 layers. Remember, the epidermis had 5, the dermis has 2. The first layer that we'll look at is something called the papillary layer. This is the most superficial layer of the dermis. Papillary layer is comprised of an areolar tissue, so an arid tissue. And found within that arid tissue are something called dermal papillae. These dermal papillae are up-like or wave-like or even finger-like projections that extend the dermis into the epidermis. Now, I have a slide here that I'll show you that. If you take a look, here is the dermal papillae, dermal papillae, dermal papillae, dermal papillae, and that's found within that papillary layer. These, sorry about that, these assist in making each of our fingerprints unique. So this is how we can identify fingerprints and make them unique. Well, it's due to that dermal papillae of the papillary layer. The deeper of the dermal layers is known as the reticular layer. It's deeper and much thicker part of the dermis, and it is really comprised of dense, irregular connective tissue. Now we have a variety of associated fibers, protein fibers, that are found within the dermis, and that's the collagen, elastin, and reticulin. Now, going back to this slide, this is a nice histological section that I urge you guys to study that shows you the difference between here we're looking at the epidermis, and then what we're looking at here is the dermis. So notice this light bluish area. That is really the papillary layer of the dermis, and it's made up of that areolar tissue. The reticular layer is this deeper, darker blue or purplish color, and it's comprised of the dense, irregular connective tissue. The reason it's going to be a little bit darker is, if you recall, dense connective tissue is densely packed together of protein fibers. So it's obviously going to stain darker. Why? There's more to stain there than what we see in that areolar tissue. All right. Really, if the epidermis doesn't have a blood supply and the dermis has a limited blood supply, then really how do these layers of the integumentary system get nutrients? Well, it's through diffusive properties. Now let's talk about that for a moment. Blood vessels are located in the dermis, but not the epidermis. But we know regions of the epidermis are living. Okay? Therefore, blood vessels allow for diffusion of nutrients and oxygen to get to the stratum basale. If you recall, the stratum basale was that deep layer in the epidermis. It contained those keratinocytes and the melanocytes that are undergoing mitosis. For those cells to actively undergo mitosis and maintain metabolic activity, they need ATP production. Where does ATP get synthesized from? Cell respiration, utilizing oxygen. So the blood vessels found within the dermis and the hypodermis are supplying the nutrients and oxygen through diffusive properties to the superficial tissues that don't have it. The minute that really diffusive property is gone, i.e. the stratum lucidium, and then now we don't really, the stratum granulosum, and now we don't really have nutrients there. Well, we also see oil-like water, barriers being created to prevent water loss. Well, this allows the more superficial cells to die. We can also discuss heat regulation with respect to the integument here. Now, I'll give you 2 examples. First example is we're going to increase the body temperature. let's just pretend like you go for a long run. Let's say, I don't know, 10, 15 mile run. Okay? Obviously, your body temperature is going to increase. What is this going to do to the vascular supply, or the blood vessels located in the integumentary system? They're going to undergo a process called vasodilation, meaning they go from, let's just say, a garden hose to really a firefighter's hose, where they have vasodilated. What this means is now, because they've enlarged, we have more blood flow there. That more blood flow has allowed now really blood to essentially be closer to the surface. Therefore, when wind or air is moving past this integument, it's going to be pulling the heat along and releasing it from the body. The opposite is true if you have a lowered body temp. Let's say you decide to go up to the mountain and go snowboarding or skiing, and you, I don't know, forgot your jacket or something. Therefore, your body temperature is going to decrease. So you get a decrease in body temperature. What this is going to do is now vasoconstrict the blood vessels found within the integument, meaning instead of going from large, they're going to shrink down in size. This is going to decrease the blood flow from those appendages, and then it's going to actually reroute it to the central portion of the body, therefore heat is conserved. So allowing, body to stay warm. All right. Couple sensory structures that we'll discuss here. The first is going to be the Meissner's corpuscle. The Meissner's corpuscle is found within the more superficial layer of the dermis, and that's that papillary layer, the areolar connective tissue layer. You'll notice here this is that dermal papillae, and what you're looking at is a Meissner's corpuscle in it. Here's an enlarged histological view of one. Here's one just lying within that dermal papillae. Here's another portion of it within that dermal papillae. The Meissner's corpuscle responds to light touch. Compare that to something called the Pacinian corpuscle. This is a much deeper sensory structure that's located in the dermis of the reticular layer of the dermis. It has this onion-like appearance when stained. So both of these are histological sections of it. And it responds to deep touch and pressure. So deep touch and pressure. You'll have to be able to identify both of those in histological sectioning. So as always, make sure you're practicing with that DVD and in lab of that. Now, we can break the integumentary glands down into 4 different types. And some of these actually are broken down further into subtypes. All 4 of these are considered exocrine glands, meaning they're excreting something to the outside portion of the body. First type we'll look at is sudoriferous glands. Sudoriferous are your sweat glands. Then we'll discuss sebaceous glands. Sebaceous glands secrete something called sebum. This is an oily substance. Then we have ceruminous glands that secrete something called cerumen, aka earwax. And then we have lactiferous glands that, produce milk and secrete milk for breastfeeding. So as I said, we'll discuss sweat glands first. Sweat glands can be broken down into 2 types, merocrine and apocrine. Merocrine sweat glands are the most numerous type of sweat gland found throughout the body. And the merocrine sweat glands are really just a long tube that is leading to really the surface of the skin. And it's what's going to excrete sweat. Now that sweat is heavily involved in cooling the body. Apocrine sweat glands are actually little sweat glands that actually lead to hair follicles. Now these apocrine sweat glands produce sweat that contains fatty acids. They become very, very active during high times of stress and actual sexual stimulation. So you see apocrine glands that get activated during stressful times and sexual stimulation, and really these are the ones that a lot of people associate pheromones with. Uh, it just so happens that a lot of the natural flora of the body, meaning the bacteria that live within the body, can utilize what's being excreted out of these apocrine glands, and that produces BO, body odor. So these are the stinky glands, if you will. Sebaceous glands are the glands that secrete sebum. This is a oily substance, and in a moment here you'll see what I mean by this flash-shaped gland with a duct that opens into a hair follicle. But really, what's the function of sebum? Sebum really coats hair and provides a waterproof, coating around it. For those of you that are familiar with conditioners, so let's say you jump in the shower, you wash your hair with a really abrasive type shampoo. A lot of the times those shampoos will pull sebum off of the hair. And this can actually damage hair to some regard. That's why companies now have put out conditioners, and those conditioners are designed to actually replace sebum. So here are just a couple of these glands in a really a nice summary. Here's that apocrine gland that's leading to really a hair shaft. It's going to, be active during stress and sexual response. Here's that flask-shaped sebaceous gland that produces, sebum that, covers hair. We then have the merocrine sweat pore. That's that tube that leads to the surface of the skin. You'll get a bead of sweat, and as air moves across, it's pulling heat and the sweat away, pulling heat away from the body. So just interesting stuff. All right. Last part that we need to talk about for this first, for this first Unit 2 lab is going to be the structure of hair. This will be really the only slide that we discuss with it, but hair has a region called the dermal papillae as well. You can see it here, and in your lab manual it has a really nice picture of it. But the dermal papillae is a region that has a lot of blood vessels and nerve supply to hair. We then have the hair bulb. The hair bulb is the actual active growing portion of hair. So you see it here. There's the hair bulb. Then the root sheath. The root sheath encloses the hair and actually begins to form a bulge. If you see that, that's the bulge that it's talking about. And what's important about the root sheath, it's the source of stem cells for the follicular growth of hair. And then, as we've discussed before, you have the sebaceous gland. That's that flask-shaped gland that's going to produce sebum to help protect hair. And then what we haven't discussed is the pileo-erector muscle, or the pili erector muscle. Just depending on who's teaching it, they may say it differently. Really what this is, it is a muscle, a smooth muscle that allows hair to stand up on end. So if you've ever, I don't know, been walking down the street and maybe you're taking the trash out of your apartment complex or something to that regard, and you just sort of get that weird heebie-jeebie feeling and your hair starts to stand up on end, that's the pileo-erector muscle that's doing that. So it's a smooth muscle, involuntarily activates, and it causes hair to stand up on end. All right, that's it for AV Lecture 2A. In AV Lecture 2B, we will be doing Introduction to the Skeletal System. Make sure you guys practice some of that stuff, and I'll talk to you soon.